My name is Jeffrey Broadbent, and this presentation will be about RED1 induction and how RED1 is able to regulate skeletal muscle gene expression signatures following a period of acute aerobic exercise. So what is RED1? RED1, or Regulated in Development and DNA Damage 1, is a protein which is induced in the body under a condition of stress. The function of RED1 is to regulate genes associated with skeletal muscles. An important note on the expression of this protein is that it is independent of mTORC1, which is an important regulatory protein that is commonly associated with ATP production in muscle cells. This means that RED1 is able to perform its function by regulating other genes that are related to the ability of muscle tissue to produce ATP without needing mTORC1. So it's important to understand how RED1 gene expression is induced in muscle tissues. So the way that RED1 expression is induced is through a period of acute aerobic exercise. This intense exercise causes RED1 to be expressed in muscle tissues. This induction process, however, is transient, which means that it is very, very short-lived. The rate of RED1 expression is highest 1 hour after completing exercise and significantly decreases after this point. The genes that RED1 then goes on to regulate produce proteins that are responsible for a number of things, like the creation of more mitochondria and an increase in oxygen capacity for muscle cells. Both of these things ultimately allow for muscle activity to be sustained over a longer period of time. Since RED1 is regulating a number of genes that all share the same common purpose of increasing muscle productivity, it is said that RED1 is regulating the skeletal muscle gene expression signature. What a gene expression signature is, it is a group of genes that follow a pattern of function and are regulated by a single genotype. In the case of RED1 is the protein responsible for this regulation in gene expression. So next we'll look at how the actual experiment was performed involving RED1 expression. So two different types of mice were used to study how RED1 regulates different genes associated with muscle function. Mice with a wild-type RED1 gene were compared to mice with a knockout for the gene responsible for producing RED1. These mice were then separated into two distinct groups within themselves. One group of mice would be serving as a control, and the other set of mice would be going through a set of aerobic exercise in order to determine whether or not a period of acute aerobic exercise would actually induce the expression of RED1. Conditions like access to food and water, as well as cage space, were controlled for between the two groups of mice. The exercise group of mice had to run on a treadmill for 60 minutes at a high speed. Following this period of exercise, the mice were sacrificed at 3 separate time intervals. The mice were evenly divided and sacrificed over periods of 1 hour, 3 hours, and 6 hours after completing the aerobic exercise on the treadmill. RED1 expression was determined by performing a reverse transcriptase PCR on samples from the muscle tissue of the mice to see if the RED1 gene was being actively transcribed. By comparing the levels of mRNA between the sedentary and exercise groups, as well as between the different time periods after exercise, conclusions could later be drawn about the induction of RED1 due to acute aerobic exercises and how those would affect different genes that are regulated by RED1. The results of the experiments using the different mice resulted in some very important findings in regards to the expression of RED1. First, it was determined that the transient nature of the induction could be confirmed. An increase in RED1 protein expression of 860% only 1 hour post-exercise shows that aerobic exercise not only induces the expression of RED1, but it does so very quickly. Second, it was also confirmed that mTORC1 expression does not disrupt RED1 expression. Also, it is important to note that the researchers were able to determine where in the cells RED1 was actually being expressed. After the period of exercise, there was a 240% increase of RED1 expression in the nucleus and a 392% increase of RED1 expression in the cytosol. This result could have implications in regards to how mechanistically RED1 is able to regulate the expression of other genes within its signature. In regards to the gene expression signature of RED1, the researchers were also able to confirm this signature by looking at the levels of mRNA expression for these genes in the mice that had the RED1 knockout compared to those with a wild-type copy of RED1. So this figure looks at some of the results of the experimentation between the RED1 wild-type mice and the RED1 knockout mice. This figure shows the different transcription levels for genes in the gene expression signature of RED1. Looking more closely at the figure, you can see that two different variables are being used for comparison. The first is a comparison between wild-type RED1 mice and knockout RED1 mice. Part G of this figure down at the bottom shows that both wild-type and knockout mice were equally capable of performing the exercise, which means that the knockout did not inhibit in any way the mouse's ability to run on the treadmill. Also, within the wild-type versus knockout variable, they were looking at mice who were made to exercise versus mice that were sedentary. There was little variation between the gene expression rates for the RED1 knockout mice between the exercise and sedentary groups, which was expected. However, for the wild-type RED1 mice, there was a statistically significant difference between the sedentary group and the exercise group for genes A, B, C, and D, as seen in the figure. This difference is denoted by the asterisk and the pound symbol. This difference also means that RED1 must be being expressed and upregulating the expression of the other genes in the gene expression signature, but this upregulation is only occurring for the mice in the exercise group, which is also as expected. So further analyzing the results seen in the previous figure, it is known that in some way all of these genes play a role in increasing the capabilities of muscle tissues. The expression of all of these genes seen in the previous figure were somehow activated by RED1 protein. While these genes all improve the muscle's ability to perform aerobic exercises in a different way, their effects all can serve the cells long term. Since these proteins cause lasting changes in the cells, it is possible that RED1 expression may have implications on the future function of the muscle cells. One gene that was looked at in particular, and is part of the RED1 gene expression signature, is ORM1. ORM1 produces a protein which is secreted by the muscles into the circulatory system. This secreted protein then enhances the exercise capacity of muscle tissue. Another gene looked at in the previous figure was JUNB. JUNB is a known transcription factor which was shown to be upregulated by RED1. The protein expressed by this gene causes muscle hypertrophy. The buildup of muscle tissue would be another example of a long-term adaptation caused by this gene expression signature. So my discussion questions for this paper are, how can the results of this paper explain why exercising becomes easier over time? And how do you think RED1 specifically alters gene expression?